In this chapter, we're going to be talking about cell division, in particular the process of mitosis. In this video, we'll discuss the different types of cell division. We'll also talk about the organization of eukaryotic chromosomes, and then we'll talk about the cell cycle. By the end of this video, you should be able to name and define the three types of cell division, to describe the organization of a duplicated eukaryotic chromosome, and to identify all of the events that occur during each phase of the cell cycle. So to begin, let's talk about the three major types of cell division. So first, we have binary fission. Binary fission is specific to prokaryotic cells like bacteria, and in this process, prokaryotic cells divide to form two identical daughter cells. The second major type of cell division is mitosis. Mitosis is division of eukaryotic cells to form two identical daughter cells. So in both of these cases, binary fission and mitosis, what we form is two daughter cells that are identical to the parent cell. So these are basically clones. Our third type of cell division, which will be the major focus of the next chapter, is a little different, and that is meiosis. So meiosis is the division of eukaryotic cells to form two non-identical cells that each contain half of the genetic information of the parent cell. So these cells aren't identical to one another, and they aren't identical to the parent. So in this case, we're not forming clones. And meiosis is most commonly thought of as being associated with formation of the gametes for reproduction. But in this chapter, we're going to be focusing on the first two types of cell division, binary fission and mitosis. So first, let's talk a little bit about binary fission. So binary fission is division of prokaryotic cells. So here you can see an E. coli bacterial cell, and as you remember, the DNA in prokaryotes is circular, and the prokaryotic cell is surrounded by a cell wall. So what happens during binary fission is first chromosome replication begins. So the DNA is replicated beginning at the origin of replication, as you've learned in the DNA replication chapter. Replication continues, and we start separating these two plasmids. So when replication is complete, we have two complete plasmids that have been fully replicated, so two circular DNA structures, and then the cytoplasm is divided and two new daughter cells result. So this is a pretty simple process. However, it becomes much more complicated in eukaryotes. So that leads us to eukaryotes. And as you remember, the eukaryotic chromosome is very different from prokaryotic DNA, which is a plasmid. So the eukaryotic chromosomes are linear, so that actually impacts how the cell will divide. So as a part of mitotic cell division in eukaryotes, the first thing that has to happen before we can even enter mitosis is that the DNA has to be replicated. So here you can see we have a chromosome, and when that chromosome is replicated, it forms the X-shaped structure that most of us are familiar with as chromosomes. But I want to talk a little bit about the parts of that duplicated chromosome. So here you see a larger version of a duplicated chromosome. So the DNA is very tightly packed and condensed using all of the steps that we talked about in organization of DNA structure. And here are the ends of the chromosomes, the repetitive DNA called the telomeres. So we talked about that in the last chapter. We also have telomeres at the other end. Remember that these are the parts of the DNA that can shorten, or that do shorten with age. In the center, we have a structure called the centromere, and we'll learn that there are proteins associated with that centromere called the kinetochore proteins, and those are going to become very important during mitosis for separating the chromosomes. So each one of these, each half of this X-shaped structure is an identical copy of the same DNA. So these two identical copies are known as sister chromatids. So this is still one chromosome because there's no new information. These are just identical copies of the same initial chromosome. So those are called sister chromatids. So what mitosis actually does is it separates those two sister chromatids. So as I mentioned, we start out with our chromosome, which becomes duplicated. So here we have our duplicated chromosome. And in eukaryotes, the process of mitosis separates those two sister chromatids. So now we have two new daughter cells that each have a chromosome that's identical to the parent cell. So we have two clones of the parent cell. And note that each one of these chromosomes is still double-stranded DNA. So we haven't separated the two strands of the DNA, we've just separated the identical copies of the DNA that were made by DNA replication. So how does this mitosis fit into the life cycle of the cell? So the life cycle of the cell is organized into what's known as the cell cycle. And so let's talk about these parts of the cell cycle. And basically, in the cell life cycle, the cell just goes through this process again and again. So first, we have the G1 phase. This is the period of growth that happens immediately after mitosis is complete, and at this point the cell grows and this is before the DNA replicates. So the cell is actually increasing in size during this time period, and in addition the cell is making all of the proteins that it needs to make before the DNA gets ready to replicate. Now at this point there are some cells that aren't going to divide again. So for example, many of the neurons in your brain have stopped dividing. So these cells can enter the G0 phase, and this is just a period where the cell is mature, it's not going to divide anymore, so it just arrests in the cell cycle. So this is for non-dividing cells. But for a cell that's going to go on and divide, at this point it enters the S phase. S phase is the period at which the DNA replicates. So S stands for synthesis because this is when DNA synthesis occurs. And at this point, the chromosomal proteins are duplicated, so our histones are duplicated and our DNA is duplicated. After this, we enter G2 phase. G2 stands for gap phase 2, just like G1 stands for gap phase 1. During this period, the cell doesn't usually grow. Instead, this is a time when the cell actually prepares for division. So it's making all of the proteins that are required for mitosis. And often in human cells, this is actually a very short time period because there's not any growth going on. The cell is just getting ready for cell division. So then we enter mitosis. So M phase is when the cell is going to actually divide, and this includes both nuclear division and division of the cytoplasm for most cells, or cytokinesis. Now, not all cells undergo cytokinesis. There are some cells, like your muscle cells, in which the cytoplasm is not divided. So mitosis frequently occurs with cytokinesis, but not always. And we'll discuss the specific subphases of mitosis in class. So this is the cell cycle going from G1, where we grow, S, where the DNA is duplicated, G2, where we prepare for cell division, and mitosis, where cell division occurs. And that's the end of this video.